that we're going to start now. So welcome everyone to this new space from the workshop of the conference. Hola. Well, this space seeks to provide training for both technical and gender perspective. In this case, today we are going to have a beautiful workshop about phylogenetic orthology inference for comparative genomics. In this workshop, Maryam and Zaide from the RSG Colombia will be exploring these concepts both from the theoretical and practical standpoint. In evolutionary research, it is essential to identify phylogenetic relationships among organisms based on their gene and predict protein content. Orthology analyses are very useful in this respect and allows us to find out whether part of homologous genes are orthologous or paratrous. So welcome to Zaide and Maryam.
Thank you, Lucy. Is it time to start talking or you have something else to present?
No, it's now your turn.
Okay, great. Well, thanks for introducing us. I think we're gonna introduce each other very quickly at first, but then we wanna hear from the participants who they are and where they're joining from and what's their interest in this workshop. And then we'll kick off with the actual course. So for that, just let me quickly share my screen. And go to our presentation here. So here's both of us who are the instructors for this workshop. My name is Maryam Chaib, as Lucy mentioned. I have a master's in ecology and evolutionary biology, which I graduated from the University of Groningen in the Netherlands and the University de Montpellier in France. And then I also did a PhD in microbial ecology and bioinformatics as well in the University of Groningen in the Netherlands. And I've mainly researched the evolution and maintenance of symbiosis in a variety of different systems, biological systems. And so that's more or less my background. I'll let Zaide presenters introduce herself.
Hi, everybody. I'm Zaide Montes. I'm from Bogota, Colombia. I'm a biologist and microbiologist with a master's science in biological science. And now I'm a PhD candidate at Lund University. And my main interests are insect chemical ecology. So I really want to know what is so important, the relation between chemical cues and how this could be relevant for the for behavior or insect biology. And I also work in the consequence of global change on insects, but mainly with the insect olfaction. This is like my general background.
I'm gonna go back to sharing my screen, and please feel free to ask any questions, raise your hand or put it down in the chat box. I think Zaide and I are gonna try to be on the chat along the course. This is more or less the agenda that we have envisioned for the day. We have a bit of an introduction to homology and what broadly comparative genomics is. Then we go deep into the subject of the day, which is orthology and phylogenetic inference. It's about 50 minutes to an hour. We go into genome-based and gene-centered methods for orthology inference. And we think gene-centered methods, which is what we're really focusing on. We're going to touch a bit on the types of methods there are. And then we're going to go into the tutorial and the example that we're going to be sharing today with you. And that's really the longest, of course, part of the workshop is the practical. We're going to have an introduction to what the biological system is, then we're going to have a little break because by then we will be already like 1.5 hours into the workshop. And afterwards, we have over an hour to be running these things together. And then we'll go back to socialize and discuss the findings and conclude. So this is more or less what we have in mind for the day. So we're just go very basic here on what comparative genomics wants to achieve. And well, basically it is centered on using an assumption that there are regions of common ancestry in different genomes across lineages that we want to use and track, and that allow us to actually compare what's these different genomic regions that are present across lineages. And we can go into that comparing either single residues in an alignment, which most of you, I'm sure, are familiar with, sequence alignment. We can also try to do this, not maybe going to the single residue resolution level, but looking to genome segments. If you have a particular region in mind that is interesting because it's a region of virulence, or for some reason it's related to a phenotype of interest that you have in your work. You can focus on a genome segment also if you have a very fragmented genome, and maybe more look a bit more at how it's structured, if it somehow genes across lineages follow the same order of genes that are present there, or it's a resolution, a level of resolution that it's a bit broader than looking at single residues in sequence alignments. Or you can really try to compare all the genes that are present across the genomes. And so for the development of this workshop, what we're going to do is we're going to focus on the genes as the evolutionary and functional units, and we're going to try to extract the whether they have the similarities between them arise either by speciation or by duplication. And so the reason why we can do, as we said, genome sequence comparison is actually based on a principle that is kind of an assumption that is cross-cutting in comparative genomics and any bioinformatics work. Indeed, it's that function conservation is the origin of sequence conservation. And the reason for this is the assumption is, and that we keep on finding evidence, experimental evidence for this, is not that somebody just made that up, but the assumption is based on, if you have a genomic region that has some sort of functional constraint that is important for the for expressing a particular function, that region is going to be much more robust to the effect of mutations. So mutations are easily going to be deleterious, and they're not going to be passed on into next generations. So that means that those regions somehow stay more conserved because they cannot accommodate mutations at any place of that genomic region. Instead, like in contrast to that, when you have genomic regions that are not...
Es un workshop que me encontré.
Sorry.
Sí, sí, es como relacionado.
Sorry.
Can you help me
Yes.
mute the participant? Thank you. But then if a genomic region is not really associated with a particular function, it can easily accumulate more mutations during the course of time. And those regions, you'll not be able to find a strong signal that relates to a particular functional domain, or a particular motif that is related to this function they're performing. So the reasons then behind to have conservation in different genomic regions are going to be if these are coding sequences or if they play a role in regulation. That's also part of the genomic regions that are more conserved across different lineages. The degree of conservation and how far or how widespread along lineages in a phylogeny you can find that, well, that varies depending on these particular functional constraints that are related to each and what kinds of functions they're related with. Because, for instance, if you have a gene family that is related to immune response, you actually, it's beneficial that these regions are able to vary quite a bit, and then those regions are a bit more complicated to analyze because of all that variation. But some others, if you have a protein binding site or a particular where you have the catalytic site associated to an enzyme, all that's going to be extremely conserved. So, but this is the like genome sequence comparison. It's based on that assumption. And then how these genome comparisons are used, then they help us identify different aspects, like what are genome structures and functions. So we not only can tell that a particular region has let's say a profile or a signature of a particular gene family that is known to be associated to a certain function, but also you can tell where genes start and end by specific patterns that are very conserved for just genome structure that has very specific constraints. And you can do that through genomic comparisons, try to identify those structural aspects of genomes. Also, those that participate in regulation, gene-protein interactions, and non-coding RNAs that are involved in regulation as well. I think somebody raised her hand, Maria Isabella.
Hi, yes. Regarding to this aspect, we can guide ourselves in order to identify these regions by the genetic neighborhood. For example, I heard about this concept that is called synteny, where we have conserved genes and the sites of our gene of interest. So my question is, we go just solely by gene specific identification, or we have to go around the neighborhood of the gene?
Right, so that was part of what I was mentioning before, that you can go about genomic comparisons at different levels. So we'll be centered later on during the example and the orthology inference. Will be using a gene-centered approach, but you also have the genomic fragment approach. And then in that case, what you're actually doing is considering an entire like contextualized genomic region where it's important to analyze the order in which these sequences are found across the genome, which relates to the concept of synteny, which we'll actually touch base on in a minute. But so, yeah, it doesn't have to be gene-centered, as I was mentioning earlier. And it depends also on your question of interest, whether it makes more sense, and also it speaks to the genomic resources that are available for your organism of interest, whether you'll go for a more like a broader approach in which you consider specific genomic fragments, or if you'll go for particular genes. But it depends on the system and the question that you're interested in.
Thank you very much.
Yes. I think someone else raised their hand, but I missed it now. Well, if the question comes back, let us know. So that's why we go then into the concept of homology, because we're basing all our assumptions in genomic comparisons on actually the fact that there are the relationship between these different lineages have descended from sorry
Put dot with that.
sorry
Hmm.
Is there an open microphone again? Okay, apparently not. So the basis to be able to conduct comparative genomics is the fact that we assume that the relationships between these characters have descended from a common ancestor. So there is an ancestral character from which differences arise in different lineages that descend from this ancestral character, but they have a shared ancestry by descent. So that's what we refer to as homology. And the opposite to homology is analogy. And that's where the characters that have converged to look or superficially are similar and may be related functionally to similar things, they actually did not arise by descent of an ancestral character, but have converged independently into something similar. So the textbook example for this, you might know it, is that the bat wings and the bird wings are actually not related by descent of a common ancestor. They independently converged to do something similar, but they do not share a common ancestor. Because bats, as you know, are from a different species, in the species tree of life. They're not sister species of birds. Birds are like reptiles, and bats are not related to them. So you have a functional character that has converged to evolve to a function that is similar, but they did not arise by a common descent. So we will not be touching on analogous characters. We'll be touching on homologous characters that are shared by common descent. And we'll be focusing within homology what kinds of relationships arise, which are orthology and paralogy. But before we do that, I thought, as is the case here, that there's a very, very broad spectrum of backgrounds, and maybe you haven't been exposed much to terminology around a phylogenetic representation. So we wanted to make sure you could follow a little bit of when we show these representations, what they stand for. And every time we do a phylogenetic representation, it's important that we're aware that what we're doing is showing the hypothesis of how these things are related to each other. And so we have an ancestral node, which is associated to the root of the tree. The way that you specify the root of a tree is dependent on which group you find as an outgroup. So anything that, let's say, is external to what you're studying here in between, everything that is descended from this node, then all of these connecting lines are called branches or lineages. The lineages is a word you'll hear me talk about a lot. You have internal nodes. So this is the ancestral. Everything in between in this region is the internal nodes, which also represent divergence points, which are the hypothetical ancestors of taxa or genes. And everything here at the right-hand side by the end of the branches are the terminal nodes, which represent the either taxa or genes that are used to infer the phylogeny. So an important distinction to make, which will be coming back to repeatedly during the workshop, is that you not only have species trees, you also have gene trees. And an important aspect to orthology and paralogy inference is how well species and gene trees kind of follow one another. And we'll come back to that, but it's important that you know that this representation of the tree can be used in different contexts. So for species and gene trees, they can be used. And so here it's a representation for a gene tree. And the reason why we know that is that, as you see down here, there are two cases in which genes for the dog come up or for humans come up. And there is a frog here. So there are 5 genes in this representation. And what you have in the star, it's a duplication event of a particular gene, which is why we have 2 times the these terminal nodes for both the dog and the human. And as you see here, this is the speciation event that eventually led to the divergence of not only dog and human, of course, this is a very simplified phylogeny. Of course, there were other lineages that arose, but at some point these share a common ancestor. So we're visualizing both a duplication event and speciation events. The first speciation event occurred further back in the evolutionary history, which was one where the ancestor led to the divergence of frog and the lineage that eventually gave rise to dog and human, for instance. And so I want to come back to the concept of homology again. Now that you are a bit more familiar with the representation of the tree. And remember that homology is all these relationships among genes that share a common ancestor. So in this example, no matter the origin of the relationship, no matter if it was a duplication or if it was a speciation event, all of them are related by common ancestry. So all of the genes in this example are homologous amongst themselves. What we're trying to dissect is not whether they arose by common ancestry, but whether the relationship they have is one that arose by duplication or speciation. And so that's where we move to the orthology definition first, which is defined as the relationship on a pair of homologous genes where the 2 genes have emerged through a speciation event. And so in that case, I would like to ask the participants, what do you think an example of a pair of orthologs would be in this phylogenetic representation? And yes, please, if you have questions, also go ahead and ask them. If you're feeling completely lost at this point, also do let us know so that we can make sure we're all at the same place to continue talking.
An example could be C1 and I can annex one.
Exactly, because they share this speciation event here in the back of the tree. That's right. And so exactly, so for instance, C1 and X2 or C1 and Y2 or X1 and Y1 are actually orthologs, which is also one answer that came up in the chat. So because the speciation event occurred here, this is why we can say there is speciation, that they are orthologous, because they arose due to a speciation event. Also up here, this is a speciation event. Then before going into paralogy, I just wanted to give you this broad overview of how gene duplication more or less looks like, or how we represent it in the view of a genome. And so you have here this protein coding DNA. Upstream the gene, we have some regulatory regions, which is control when and where the protein is expressed. And say duplication of genes occurs, and this gene duplicates into two identical regions, which actually is not. So you see in this example, you not only get the duplication of the gene itself, of the protein coding gene, but you have the duplication of the regulatory regions associated with it. In that case, what can happen, so that mutations that arise can occur both at the regulatory region level or also within the protein coding region. And one thing that can happen is that if the regulatory region does change, say the function of the gene ceases to occur the way it used to. And this can be evidence in the way it expresses or in the conditions under which this region now is expressed. But also you can have the situation in which the coding sequence also changes, and then they have a different function than what it, like the original copy used to be working for. And so this is more or less what we're trying to detect when we are looking for paralogy. And so paralogy, just to give you the textbook definition, is the relationship among a pair of homologous genes, that has arisen through gene duplication. And then can somebody comment on what are examples of paralogous pairs here in this representation? You got them right with the speciation. So yeah, so Sophia is saying Y1 and Y2 are examples of duplicates. That's correct. Also X1 and X2. Thalia said they can't activate their audio. Can you please turn that option on? So yes, so basically just to recapitulate, in any pair of genes that arose by this duplication event, either X1, X2, Y1, Y2 would be duplicates in this example. There are a bit more instances of scenarios that have an associated terminology that I wanted to quickly touch base on here. One of them is xenologues, which are genes that are shared into species by horizontal gene transfer. This is somewhat detectable within these kinds of analysis, but you wouldn't immediately call them horizontal gene transfer events. You would actually just see them as, like, the author of OrthoFinder calls these like phylogenetically misplaced genes, but the series of analysis that you have to do downstream are quite varied in order to try to support whether a horizontal gene transfer event occurred or not. We could talk about that later, but in this case, these xenologues, you would at least first see them as like phylogenetically misplaced. But that occurs, and that's the term associated to those kinds of relationships. Then you have out paralogs and in paralogs, and the out paralogs are just paralogs of a duplication event that arose by duplication, but this is before the speciation event of interest. And in paralogs are paralogs of a species-specific duplication event that follows a speciation event of interest. And something that you'll see a lot in orthogroups, once we have done the orthology inference work, you'll see that you have several in paralogs in a clade following a speciation event of something that you can call an orthogroup. So you'll see a lot of in paralogs in the analysis, for instance. So anything that arose by duplication after a particular speciation node of interest. And so you can observe this in a gene phylogeny. So remember to have this clear distinction that there are species trees and gene trees. In these cases, what we're observing are genes in a phylogenetic representation. And so when we have in a node here by the root is the ancestral gene by which all of the different copies then arose. What we're trying to determine is what occurred in these internal nodes, whether it was a duplication or a speciation event. And so here in this case, first, you got the emergence of two genes A and B by a duplication here in red, and subsequently you got the speciation event. So the speciation event is shown in parallel for gene A and gene B because all of them in the different lineages. In this case, we have Arabidopsis thaliana and Oryza sativa in this representation. They gave rise, so by the divergence of these two species, you get that all these copies also diverge, all these different genes. And you have then duplication events that occur specifically within each lineage, specifically within Arabidopsis and specifically within Oryza sativa. And also in the case of gene B, you get that there is no internal duplication for a particular gene for rice or the species related to rice, but you did get it for Arabidopsis. So not all genes following speciation may have duplicated. That might have happened for some gene families, for some others not. And just if you have the speciation event here in the middle, what you see, all of these are orthologs because they arose by speciation, but then if they suffered later on a duplication event, which is basically all these three events that we have down here at the right-hand side of the tree, they are specified in the circle in the green color and they're in paralogs. So what I was explaining before. And this is, again, just try to recapitulate on the same concept because these are the relationships that we're going to be identifying today in the workshop. We need to make sure that you truly understand what the terminology is, in which cases, for instance, here, these two are xenologs because there is a lateral gene transfer event. When two copies are out paralogs or in paralogs, depending on which one is the speciation event of interest, and just anything that has diverged by a speciation event, such as A1, B1, or A1, C1. You need to make sure that you understand this concept. So please do say something now if you don't, because that was like the basics of the concepts and the terminology. And now we're gonna move on into the techniques by which you can identify them. Okay. It looks like it's clear. And so what happens with copies that emerge by duplication is that they can have three different fates related to how they perform later on. Yes, Claudia.
Sorry, I just wanted to make sure in the last picture, there were 3 C genes. Yeah, that's right. So they would all be in paralogs, all the Cs, C1, C2, and C3.
No, so C2 and C3 would be in paralogs because they emerged by a duplication event that occurred after this speciation event.
But the relationship between C1 and C2 and C3?
So they emerged in this duplication event up here, number 1. And the lineage that led to C2 and C3, this copy duplicated later after a speciation event that occurred in between both duplication events.
But they would all be paralogs, only not in paralogs?
They would all be paralogs, but C2 and C3 would be in paralogs after the speciation event number 2. If you consider speciation event number 1, they would all be in paralogs after speciation event number 1. So there has to be a speciation event of reference to be able to denote in a certain way out or in paralogs if they are relative to a particular speciation event. Sorry.
Oh, okay.
Is that clear?
Yes.
Cool. And so duplications, there's one assumption that is important to consider functionally and at the level of sequence identity. Genes that emerge based on following a speciation event tend to be more similar amongst each other than genes that resulted via duplication. Genes that resulted via duplication are assumed to have a higher divergence because by genomic constraints and say energetic efficiency, things like that, it is assumed that multiple copies of the same gene are not going to be like tolerated within a genome, especially also in prokaryotic species. And so what happens with these copies of the genes is where if they evolve fast and start performing different functions, then that allows for these copies to be kept in the genomes. And that also means that this is represented at how, by how genes, if you align them to each other, how we see divergence among them. And so that's why we also have this like well-known fates of paralogous genes. One scenario here is where there's non-functionalization, where because this copy is not being used to produce, for example, a certain gene product, it accumulates a lot of mutations, eventually yielding that gene non-functional. And that's the origin of pseudogenes, if you come across that concept. Then neo-functionalization is the event in which the second copy is selected for because it evolves a new function that is unrelated to the function that existed for the original copy. And subfunctionalization is when both duplicates start being used for the same function as the original had. And a good example, well-known example of that are the different alpha and beta chains of hemoglobin. They are now used for the same function, but they are different domains, but that arose by duplication of an original copy. So these are the different fates that these duplicates have. And so the way we get at comparing these genomes, you can have a genome-centric approach and a gene-centric approach. And for instance, where you have Paulina, yes.
Yeah, I have a question. How can we identify in the sequence if a gene is in the way of selective pressure, or it has been selected, or it's not available, this information?
It is available with different metrics. So there's something called the rate of synonymous to non-synonymous mutations. Those ratios are used as a proxy for selective pressure, which is something I'm not covering here, but that's a way that you can identify loci under pressure. But what you can see, let's say, just through the alignment itself, is how diverged copies are. And the fact that a gene has evolved a new function most likely speaks to the fact that at some point it has been under selective pressure. But in order to say that right now it is under selection, there are a number of approaches, but let's say that one of the ratio of synonymous to non-synonymous mutations, it's like a widespread and well-known one. Okay, cool. So I remember someone talking about working with Mycobacterium. And that's a great example of genomes that have a very low rate of divergence, even genomes from different species of the same Mycobacterium genus. They are actually quite similar to each other, which is a very different scenario, for instance, for enterobacteria, such as those as E. coli, like the Escherichia and the Shigella genera. They, for instance, let's say they share their 16S region completely and exactly the same copies, but their genomes are really extremely divergent, even within each genus and within strains of the same species in those two genera. And that's something that is very different to the divergence rate of other species and other genera, such as Mycobacterium. And the reason why I bring it up here is that you can have global alignment of genomes in cases such as the genomes haven't diverged at a very high rate. So you could imagine these types of analysis where you're really trying to see structurally how well two genomes match each other. This is the type of analysis that is very, very handy for species in general that do not evolve very fast. You can do these types of analysis both at the nucleotide and at the protein level. And the type of analysis you'd perform would be based off how divergent those genomes are. So if you're comparing strains of the same species of bacteria, you'd want to use nucleotide level to give you enough resolution to detect differentiation across the genomes. But if you are trying to analyze different species of bacteria or different genomes in different genera in the same family, you'll want to go for the protein level, which gives you enough level of resolution for those more divergent lineages. And so this is an example of a software that is called Mummer. It's already quite old, but still very useful for those kinds of scenarios where you want to do global alignment. And that is a genome-centered approach to comparing a set of genomes. And basically what you're trying to detect in these plots is anytime you have identity, you'll observe that in the diagonal, basically each position in genome A has the same position in genome B, and these regions are perfectly aligned. And then any structural variation you'll observe by differences in how these the diagonals show up in the plot. So any inversions, which basically mean that you have totally changed the relationship of the position of B in A, you'll observe as a completely crossed, like counter cross diagonal, which is this line here in red. Insertions, which is this example here, you don't see them in any color. What you see is that this region exists in genome A down here in the x-axis, but it has no corresponding position in genome B, which is why in the y-axis there's no movement, let's say. So you see them as a cut in one of the axes, depending on where this genomic fragment is inserted. And you also can see translocations just by slight movement in either the X or the Y axis according to how these have been translocated relative to A or relative to B. So that's the kind of information that you can get from these plots. And this is how it looks for two species. Here's an example on Sanctomonas. And you see that the genomes are relatively well conserved, but there are regions of inversions that are, let's say, quite marked in this comparison. So that's one approach by which you can compare genomes that are not highly divergent. And you can also visualize, this is a software called MOV, where you can have multiple alignment of conserved genomic sequences, where you can observe if some of these regions have been rearranged, and you can also see whether these regions have conserved blocks and how the order of the sequences is like relative to one lineage to another. This is also a good way to visualize synteny, as one of the participants was talking about before. And you can also say, depict this synteny basically on how several regions align together or are present or not in different lineages across a particular clade of interest. And you can color these by different, like they can have different colors based on their predicted functional associations, that kind of thing. But say, I don't go deeper into this because we're not, like what we want to do is get to the part of orthology inference and paralogy, but that can certainly be the focus of more discussion, or maybe another workshop later on. And then so we're now really moving into the gene-centered analysis for orthology inference. We have 3 types of methods. I think, did someone want to talk?
Yes, I just wanted to ask what software that was, the one from the previous image.
Escapes me right now. Let me get back to you on that. Yeah, I
Thank you.
Can't recall the name right now. Okay, but I can also maybe later send you a good book chapter where the latest software is summarized, where you can also visualize synteny.
Thank you.
That is not only MOV, which is kind of quite old by this stage, but it's good because you can install it in any operating system, which is I think one good reason why a lot of people still use it. But there's definitely a lot of newer software that also support having a larger number of species represented, and also more complex regions where you might not have like a very complete genome, but highly fragmented genomes. Those things have also somewhat evolved a bit. So yeah, we can send you that information later. And so for gene structure analysis for orthology inference, we have two types of methods that are based on graphs and methods that are based on phylogenetic tree reconciliation. And tree reconciliation is, remember that we've been talking about having trees representing gene hypotheses on how they have evolved, the genes themselves, and how species have evolved. And the thing is, not all genes follow well the species representation because of all the duplications that have occurred, gene losses following duplications, following speciation events. And so what you would want to do is have the genes and the tree species and the species tree, reconciled, like somehow you try to infer what happened to those genes, their evolutionary history, when you map them over to the species tree. And that's when you take into account the phylogenetic component into producing these orthology relationships. And there are methods by which you can combine them both, and they're called meta methods. We sent you this book chapter by Altenhoff and collaborators, which is rather recent. And this is where they describe very well in detail of these different types of methods. I don't know if you had the opportunity to check it out, but if you didn't, that's where that information is also found that you can touch base on later after this workshop. And I'm going into tree-based methods first because the software that we're going to compare in the workshop are based on graphs first. But these tree-based methods also exist, and it's important that you know about them. So again, as you know, you have the gene tree and the species tree. In this case, you see that there is a different color and a different shape for each of the lineages. And you see that the species tree and the gene tree do not have the same topology. In this case, the gene that is identified from the lineage of the frog does not share a node with the human, which is what is depicted in this gene tree. As in the species tree, you see that it would be the gene that is associated with the dog. So the idea that what you want to achieve is a hypothesis of what happened in the evolutionary history of a gene that yielded this pattern, and how can you reconcile the representation with the species tree. So we know that the topology, the right topology for the species tree is this one, where dog and human are more related to each other than to the frog. So there would be a speciation event up here, a speciation event down here, and you'll find that early on there had been a duplication event here in the star for a particular gene, and then this gene got copies due to speciation. And so the reason why we don't find other copies of these genes in the gene tree is because there have been losses of the copies that arose before. So these are depicted by the crosses down here. So this is essentially a way to say the reason why this gene tree doesn't follow the species tree is because we hypothesize that these are the relationships that led to that. So this full representation basically conveys not only at which stages the gene got copies due either to duplication or speciation, you also get to show why, like in which instances there have been gene losses associated to a particular gene of interest. Yeah, so that's more or less the idea behind those. When we talk about OrthoFinder was originally a method that was based on graphs, but in its extension a couple of years ago, they went all the way to introduce the tree-based method to complement the graph-based method that already existed. So I think you can say that this is nowadays more like a meta method, and we're going to be talking about it in a sec, but just to know that I'm going to show you a little bit of how OrthoFinder works, and well, just to keep in mind that this kind of language is going to come back then. Sorry, just let me drink a bit of. So now we go into the graph-based methods, which consists of two phases. A phase in which you actually build the graph based on, say, similarity. And then you have a second phase in which you cluster the different parts of the graphs to build what would be the orthologous groups. And so in the first phase, the most basic method to go about building the graph is you take first all pairs of genomes at a time, and you compare all the similarities of all the genes in a particular genome to all the genes in the second genome. And you do this sequentially for all pairs of genomes that you need to compare. And you use the similarity of these comparisons as a proxy for detecting how closely related these genomes are. And you'd use something like the best hit. So the highest similarity score or the highest scoring match in the other genome is something that you would that would constitute this best hit. And just let me go here for a second. People used to use BLAST for this. Nowadays, I think Diamond is more used because, of course, is very, very much faster. But basically, you would use that to compute the best hit and actually the reciprocal best hit, sorry. So in this case, when you talk about reciprocal, it means that the most similar sequence to this one I'm comparing is going to be that one in genome B. And also, when genome B is compared to genome A, you also get the best hit to the sequence in 1A. You want to consider the cases in which that's true, because a non-reciprocal scenario is that where you have your gene sequence in 1A and the top hit is that in 1B. But when you compare 1B to either to the genes in A and in 1 and 2, you get that you have high hits that are equivalent both in 1 and in 2. And in that case, you can say that the hits are non-reciprocal. So you want to get the reciprocal the best hits in this approach. And then the second phase, which is clustering, you can have different approaches to it. One is, which is the first one that emerged, is the clustering of orthologous groups, in which you connect 3 nodes as in a triangle, and you try to go and merge triangles which contain all of the nodes in these triplets until you can no longer merge more triangles. And that's going to be a subgraph that is going to consist of your group of orthologs. You can also have something called a Markov walk into the graph and compute sequentially what is the probability that two genes belong to the same cluster. And only for those that share the highest probability, you get to put them together into the same group of orthologs. Or you can like have a graph-based approach to identifying fully connected subgraphs to say that the orthologs that you identified in the first stage actually constitute a particular group. So one is the phase where you just compute the similarities of all the sequences, all the gene sequences across genomes pairwise, and you say these are potential orthologs, and then you want to build groups of orthologs that somehow cluster together orthologs that are hypothesized to belong to the same group. So those are the two stages by which you build the orthology inference. So orthology is a non-transitive relationship, which means when, like, if you've ever heard the concept of transitivity, when you say like the outcome of A and B is the same as B and A, or sorry, A and B. If A and B are related and B and C are related, then that means A and C are related. That would be the transitivity property. But in orthology, that's not the case. That A and B are related and B and C are related does not mean that C and A are related. That's why in this computation here, you want to have only reciprocal hits because of the non-transitive relation in orthology. And so that's why you want to find also these fully connected graphs or somehow those clusters to partition them in groups that actually do share that orthology relationship. And basically what you get out of the first part of the inference is for the comparison of organism A and organism B. For instance, you have, this is an example of E-value, which is not used in OrthoFinder, is used in other approximations. But this is, say, a significant score that you get when you use BLAST. So you're gonna get for each pairs of proteins, what are the expectations values associated to each of the comparisons of each pairs of genes. And well, I don't think we need to go into all this depth, but one of the difficulties with the basic approach of using BLAST is that when you use the best BLAST hit, you're missing out on detecting relationships of one to many. Basically, maybe your gene has the same match to different proteins in another genome, that's one to many. But also you have the scenarios where both in genome 1 and 2, you have the same or very high expectation values to many genes in A and many genes in B. Those are the many-to-many relationships. And those things are not accounted for in the basic approach of the best plus hit. So other people have moved away from using a best plus hit scenario as a proxy for detecting orthology, using things like maximum likelihood estimates of evolutionary distance. In that chapter that we shared with you, there's this table of with an overview of graph-based orthology inference methods. So this is, say, the basic one. We're going to talk a bit about ortho-MCL as well, but there are other methods that are well discussed in there, and we won't be using today. But you can check, let's say, the details in there. One that is in Paranoid actually is an expansion of the best plus hit approach, but accounting for those caveats that are mentioned, the many-to-many relationships and the one-to-many relationships, and uses an outer group. So you can specify in advance when you input your genomes into the analysis. You say the genes coming from genome X, it's actually the outgroup to my analysis. And that's nowadays always a good practice to include an outgroup. And say this is a software that was also actually quite often used in Orthology Inference. And oh, sorry, I thought it was a question. And also OrthoMCL was one that had specificities and sensitivities above 80%. Only these two software, say from the classical software, are actually like good options to consider for orthology inference. And now maybe you want to have a 5-minute break before we go into the details of the algorithms. I think maybe it's been a little bit long. I could use a bit of water, maybe 5-minute break. So let's start again. And I just want to hear, maybe let's give a couple of minutes for people to come back. I want to hear how you're doing because it can get very dense. So if some of you have comments or questions by this point, please feel free to bring them up. And well, as we talk about that, we'll let other people join again. Don't be afraid to talk. The idea is to make this accessible to everybody, have you share your opinions, how you're doing with the learning, maybe for people who haven't done their masters on something like this or started it. It's a bit heavy, but I think this is the heaviest part. There we're gonna get to a practical. It's okay if you don't follow everything as well, as long as you're learning something, getting something out of it, then I guess that works too. But it would be nice to hear some of your opinions so far.
Hi, I do have a question. Maybe it's kind of dumb, but you mentioned the word proxy, and I wanted to know what do you refer with that?
I mentioned the word proxy, yes. So since we want to get what are the most related sequences, like what are the most related sequences, that is hard to measure like exactly, because there are different processes that occur, different divergence rate that occurs. And so we use a measure that we can compute to talk about that relatedness or that closeness, phylogenetic closeness. So we have to use some kind of measure. And the best measure we have here, or the most used measure we have here, is similarity. Basically considering how many of the positions of one sequence compared to one another are similar to each other. I didn't use identity because particularly when you compare protein sequences to one another, you have two ways to talk about how similar they are: the actual similarity score and the identity score. The identity would be for the given length of these two sequences that you're comparing, just all the positions, the individual positions along that region that are exactly the same in both sequences. Similarity, on the other hand, if you have amino acids that are from the same group, say they're all hydrophobic, they are more similar to each other than hydrophilic and hydrophobic amino acids, or polar and non-polar, or However, they are grouped based on their biochemical properties. Those, so the mutations in the codon in the nucleotide sequence that lead to that amino acid are fewer or easier to occur in evolution than for amino acids that are not from the same biochemical group. And so those substitutions that occur between similar amino acids, those that come from the same group, actually get scored higher. They're not computed the same as a substitution that is like completely different amino acids. So you have this measure of similarity that speaks to how closely related they are, but you're not getting like the truth, you know, you're making an inference on how they evolved and how they diverged. So that's why I use this word proxy. It's just like a way to measure something that is far more complex to actually dissect. I don't know if that answers your question.
It has actually cleared a lot of my questions. And one of my troubles was that I was comparing actually proteins that were really far away. One that was a mammal and then a fish. So the identities gave me like a broader scope on how to compare them because, sorry, the similarity, because identity was really
low.
Small, really.
Yeah. So when you want to compare things that are so distant, and maybe Zaide can help me complement here since you work with a rather divergent and broad group, you want to make sure you include other lineages in between. But Zaide, if you want to comment on this.
Yeah, that is one of the options. And another is like you can include more data of not related species because you want to just like to force or to try to create like a pressure in the analysis just to know if how related are your data. Like this is like an option. You can include more data of not related species, or you can add more data from the same species.
So no, I think if you have say 2 species in each and you have fish and mammals, you definitely want to broaden your sample. And even if the species you're interested in, like you really do want to sample broader in the phylogeny, because then you're only considering say 2 or 4 data points, and you're not going to get enough resolution in the analysis to indicate where really the speciation events occur or the duplication events occurred. So you want to include other species that are not the ones you're interested in, as Zaide was saying, to be able to have more confidence in which events you're inferring that occurred. So you should not input into your analysis only species A and B of fish and species A and B of mammals. You really want to include other genomes or other predicted proteomes better from a lot of clades that are in between those two and an outgroup as well. And that will really strengthen your analysis.
Awesome. So it would be a good idea to make a species tree and then a proteome tree in order to compare that, like as a guide.
Yes, so, well, I'll get to that in a second with the OrthoFinder explanation, okay. But you don't have to build a species tree yourself because that's a lot of work, but there are species trees available in which you will be able to, like already published from people who have made a lot of effort to include multiple loci to create a robust phylogeny of a particular, say, vertebrates, right? And so you can go there to the published literature and say, this species that represent not only mammals and fish, but also reptiles, amphibians, whatever, have published genomes that are complete, well annotated. You maybe use model species within those groups and integrate a few more genomes, like sampling along a phylogenetic gradient. But you don't have to do that work yourself, because that's another thesis. You can use what's already published.
Great.
Daniela, yeah, you're welcome.
I have two questions. The first, last year, I obtained the orthologous genes between different cultivars of Vitis vinifera, but I don't know if it's correct, my methodology, because I use the mRNA sequences, nucleotide sequences, and not the protein sequence. I made an alignment, a bidirectional alignment between these mRNAs with blast N, and the next I use a needle, the global alignment, to has the orthologous genes between all the species. So my first question is, it's correct, this approximation, to obtain the orthologous genes, or I need to use the proteins?
Because you're using different cultivars of the same species, you can use nucleotides, actually better, because that will be giving you resolution, higher resolution, because you'll get the specific mutations that might not be represented in the proteins, because you know of the degeneracy of the code to actually produce the amino acids. So in that case, you do want to do the analysis based on nucleotides. We use proteins when you're comparing much more divergent lineages.
Okay, and my second question is, well, I use blast end and needle, but my PhD advisor recommends me to use the OMA. But I don't know what is your experience with OMA, because I see in the previous slide you show the difference program to obtain the orthologous, and the next slide you show the false positive and false negative numbers, but I don't see OMA. So I don't know if OMA is correct.
That was an old paper from 2007. OMA is more recent, but Zaide might have better insight on this.
Okay.
Okay, so I have never used OMA before, but I know that it's really similar to OrthoFinder. So it's like it could be like a great idea just to test. But if you use OMA or OrthoFinder, it will be really similar to results. So, but I don't have experience with OMA. Maybe we can just check in details, because, but it's similar.
Okay, but if in my input data, for example, I need to, for example, if I use ortho MCL, I need to have one gene represents the group, or I can have the, for example, the difference transcript variation. Because in the mRNA data, I have the different transcript variation, type 1, 2, 3 for the same gene. And maybe this information can, I don't know, difficult in the parallel
those are basically isoforms of the same gene.
Related. Yeah, you need to include other data, like, because you are like you are not established relations with things that are not, all the things that you are comparing are really close.
Right, so different things there. OMA it's really a great method. The only reason why it wasn't in that picture is because it's newer, and the developers of OMA are really leaders in the field of orthology inference. So I highly recommend you use OMA. The reason why we're not including it in this workshop is because we personally haven't had the chance to use them in our projects. but OMA, it's really a great tool. So you should definitely explore it. The other thing is when you're using mRNA data, you have to think about having the same region in the genome producing these different isoforms, which are not duplicates or orthologs. It's a different kind of relationship. So you do want to have a representative of the isoforms in the analysis, because otherwise you're going to be confusing the analysis with your input for sure. Yeah, so those are the things I wanted to comment on your question.
Okay, thanks.
Talia.
Hi, I have a question. You have already showed a list of programs. So how can we decide which is the best program for our data?
So there are papers where they do benchmarking of the different software. The problem is that the benchmarking is usually done when a new software is released and is done by the authors of each of the tools, right? Those different benchmarking, the different ways of benchmarking have shown that OrthoFinder really does, like, it's highly accurate in the orthologous groups it infers. Also, OMA is quite good. The difference is that OMA doesn't go to the extent of using the phylogenetic information to produce the orthogroups based on tree reconciliation. That's like a very recent addition, and maybe that's not where you want to get. If you only want to do the inference of the orthologs and the paralogs, that step is pretty well achieved with either of them. OrthoMCL is a good tool, is old. The thing with it is that it may be harder to install, and it's also slower.
Slower, and when you use like a really high amount of data, at the end you will the software it's not able to create relations because it works based on the size of the data. So if you are using, I don't know, more than 3,000 sequences at the same time, OrthoMCL it's not able to create any kind of relations. So this is also a problem with OrthoMCL. So you have a very big data, don't use it.
Okay, thank you.
And it's also depending on your level of expertise with command line. So we're going to show one alternative to OrthoMCL, which basically uses OrthoMCL behind, but they've introduced slight modifications in the software that performs some of the in-between steps to speed it up, to make it more efficient. That's OrthoVen, which is also implemented like as a remote web server, like you can do it online. It has a limit on the number of species you can use, but it's a good option for a first exploration. It's based on a good algorithm that has had years out there and a lot of people have used. But maybe if you're new to all of this, and you want to focus on the interpretation and see how your data is behaving, you can use that as a first step. The other thing that is good practice is to compare anyway the output of several options, because you can see how robust the orthogroups that you find are to the different, like to the differences and nuances of the different methods. So that's always something in research that you don't want to like use the first thing that someone tells you about, but you want to see how robust your results are to these different methods. And in that case, for those orthogroups that are really consistent across tools, you have higher confidence on those, right? So that's also something to consider.
Okay, thank you.
Okay. Cool. So Gabriel, go ahead.
I'm sorry, guys. There's literally an airplane in my neighborhood right now, so I don't know if you can hear me. I was
I don't
saving
hear
this
the
question.
airplane, no worries.
Yeah, that's literally an airplane. I was saving this question for the end because I don't know if you're going to talk about it at some point. But in my case, I'm trying to compare whole genomes of very closely related species. So how would the procedure work in this case? Because I don't know if it would be convenient to work on orthologs in this case, since we expect most of these proteins to be orthologs.
That's true. I know someone who had that problem and the orthology groups did not help. What are you trying to detect exactly?
Well, I'm trying to get evolutionary distance between species and how long ago they separated, this type of question. But it's for a section of a genus, and we're using, of course, a few species from outside of this genus. But they're very closely related. So I think it would be adequate to use whole genomes because for being so close, I'm afraid of introducing any other bias to this analysis.
Yes, I don't think the orthogroup inference would be the way to go for you. There are approaches to whole genome alignment that you can explore. And also maybe you can focus on looking at like nucleotide level differences in regions of interest. But definitely, I don't think the orthology is going to help you here. That maybe you,
Okay,
you can send us an email later, talk about that more. But yeah, I don't think this one would be the way to go. Aside, do you have some other thoughts? No. Okay.
Sure thing. Thank you.
Okay. So we'll leave you our emails at the end. Let's go briefly into the algorithms. So OrthoMCL is one we wanted to mention because it's the classic one. And as I was mentioning before, we're going to be using an extension of OrthoMCL, which is basically a more efficient version of it. It performs clustering at the genome scale, and it uses the blast all versus all approach to identify similarity. And the reason and the fact that it uses that approach, and any software that uses that approach, basically has the characteristic, maybe a caveat, depending on your question of interest, that it is able to identify only the recent orthologs and paralogs. And that's because it uses this best hit approach, which basically gives you only the highest similarity across, like, comparing pairs of genes at a time. And so all these arrows in gray here are relationships that exist from the different copies of the genes in both species A and B, but that you may not be able to detect necessarily if these orthology relationships date a bit back into the past. But you are very well able to detect the in-paralogy relationship of the duplicates that occurred after the most recent speciation events and the most recent orthologs. And basically this, what you're seeing here is, say, the classical algorithm. What changes in particular for OrthoMCL is the way that the clustering of the orthologs is performed. I told you that there were different approaches to identifying orthogroups. What OrthoMCL does is this Markov walk around the graphs, but everything else that occurs in this flow, which is basically obtaining or predicting the protein sequences from the organisms of interest, performing this comparison via local alignment of each of these proteins to each other, the all versus all approach, in this case at the protein level with blast P, gives you metrics both at two levels, between species and within species. And the reciprocal best hits give you the supposed orthologs, and all the reciprocal similarity pairs give you the recent paralogs. This yields all these comparisons of each other all to all, gives you a similarity matrix, which is normalized by species. That means it's corrected by the size of its genome. So the size of each genome in this case is represented by the number of predicted proteins. You want to have a normalized measure that corrects for genome size. And that represents in the metrics of the similarity matrix, like hypothesized orthology and paralogy relationships. And the second step, as we mentioned from these graph-based approaches, is going into identifying the groups of orthologs. So that's basically what the algorithm is, that is how it occurs. And then as I was explaining to one of the participants before, and I'm sorry because I'm terrible with names, OrthoVent is actually a re-implementation of OrthoMCL, or it uses OrthoMCL, but some of the key, just not the key in the sense of how it works, but in how long it takes, has some slight variations of the MCL algorithm based on that, but it is an implementation of the MCL algorithm. Basically, oh, I see that I made a mistake and missed, oh no, I'm sorry. This is present in the supplementary material of the paper where OrthoVent second version was introduced. So you can go back to this exact figure in that paper, and I apologize, I did not see this error before. But basically what it tells you, and you can focus that down here, is that basically you have almost the same number of clusters that come back from the analysis that contain roughly the same proteins clustered into those clusters. So that's what I was telling the participant before, that you can use this approach of OrthoVent too online if you want like a fast version of OrthoMCL. The difficulty with it, as I was saying, is that it only supports online up to 12 species, but there is a standalone version that you can download. And so if you want to use OrthoMCL or a version of OrthoMCL that gives you roughly the same results, you should be able to use this like confidently. Okay. There is some parameter value that you need to pick for OrthoMCL and also OrthoVent, because it's based on OrthoMCL, and it is the inflation value that basically gives you the granularity at which you want to cluster your proteins. And so the default value for this in OrthoMCL, they give you a set of parameter values that is good to explore. In OrthoVent is 1.5. In OrthoMCL, they give you a series that started at 1.4, went up to 2, 2.5. It is a good default value, but based on personal experience, I suggest that you try different values of inflation, different granularity thresholds at which you want to cluster your graphs. Because some of these, depending on your study system and the rate of divergence among the groups, you will want to have smaller groups, which you achieve with higher inflation values, or larger groups, which you would achieve with lower inflation values. And so also when you use OrthoVen online, you need to specify this inflation value, or if you use OrthoMCL or OrthoVen locally installed in your machine, you can also, you need to, you cannot run the software without specifying that, or you would use the default, but it's always good to explore, just as with the different software, it's good to explore these key parameter values within the same program, because you want to see which orthogroups are also robust to different values of the same parameter. And then this is, unfortunately, they didn't have a figure this explicit on the concepts of ortholog and orthogroup in the papers. They have two papers, one from 2015, where they published the first version of OrthoFinder, where you find the details of the graph-based method that they devised to predict orthology and orthologs and paralogs. And then they have a second paper from 2019, where they extend their method to become more like a meta method, where they complement what they produce with the graph-based method to include everything that was based on trees and tree reconciliation. I told you we would come back to that. But they didn't have a good representation of these two basic concepts of their work in those papers. He did have it, though, in one of his talks on YouTube, and I have the link down here. We're going to share these slides later, which is why you see that there's a cut of his, a snippet of where he was talking about it. I just say very honestly that that's why I included it here, but you can watch the entire talk and see that this is where I got this image from, where basically you have very clearly here three specified orthogroups, orthologous groups, one for vertebrates, which includes fish, tetrapods, which have 4 limbs, that gives the name to that group, and you have 2 tetrapod orthogroups, because based on a duplication event that occurred here, you have an orthologous group up here and a second orthologous group down here. So there is some difference between what an ortholog is and an orthogroup is. The orthology is really based on 2 species, the genes in 2 species that descend from the same last common ancestor, LCA. And the orthogroup is an extension of this idea of the comparison between 2 species to multiple species, as you observe in this tetrapod orthogroups cartoon, which is the groups of genes that descend from a single gene in the last common ancestor, but for more than 2 species. So I wanted to make very clear the distinction between the 2 concepts, but unfortunately they didn't have the figure anywhere else, so that's why I point you to it in the video. Yeah, but yeah, so that's it. And so the OrthoFinder algorithm, and this is something that you can find in the 2019 paper, basically this is all the second part that OrthoFinder 2 added to the software. So in the first version of OrthoFinder, you would have the predicted proteomes for a set of species and do the first prediction of the orthogroups, which was basically out of using a version of BLAST that they developed, or they extended, let's say, the BLAST algorithm to produce based on the comparison of all proteins versus all proteins in all these different species to produce unrooted gene trees. This is what the software they developed, Dendroblast, does. And they go through a series of steps to root their phylogeny, their trees, to produce these rooted gene trees. The reason why they want to yield a rooted gene tree is that they use all the orthologous groups devised from the different gene trees to infer a species tree. And so remember this important distinction between gene trees and species tree. So in order to implement the entire second part of the OrthoFinder 2, they needed to be able to reconcile gene trees with the species trees. And so there are two ways in which they feed the inference of the species trees based off the proteomes. So they don't ask you to build a species tree yourself. They infer it in the software directly based on two types of orthologous groups based off their genes analysis. First one, they used the single copy gene orthogroups, which are basically composed of those proteins that are present in single copy in each of the genomes that you input into the analysis, which is why they're called single copy orthogroups. And these single copy genes are also used in other types of analysis. For instance, when you're deriving genomes from metagenomes, or when you are assembling full genomes, what you count to be able to say whether your genome is complete or not is the single copy genes present in the genome. So they have a good phylogenetic signal. They use those on the one hand, and they also use like a consensus of the trees that they obtain based on orthologous groups that they obtain not from single copy genes, but from genes that arise by multiple copy. Of course, those are the ones who are subject to other kinds of evolutionary events, but they're needed to produce like a more robust species tree inferred directly from the proteomes that you input into your analysis. So basically, the second stage of the analysis starts by resolving the gene trees, which is a way to say that they implement a gene and species tree reconciliation, where they are able to map what was the process that occurred at each of the different nodes. Yes. And so basically that's what yields all the orthologs and orthogroups for each species compared to all the others. We're gonna go through the output of OrthoFinder in a minute. So I really wanted, I think it's clearer to show it directly in the terminal, and all the comparative genomics statistics that are the output of this software. And then one last thing that I wanted to touch base on, Claudia.
Yes, I wanted to ask you if it's correct to use that species tree that comes from a reconciliation of gene trees as a species trees, like if your goal is to have a species tree and not orthogroups to analyze, if you can use that species tree by its own.
Right. I would not recommend it. It's a good proxy of a species tree for the efforts or the question in hand, which is orthology and phylogeny inference, and also estimating where in the species tree different events of gene duplication and loss occurred. If that's your goal, this is where that tree would be used. But if you only want to get the species tree, you would get a much more accurate tree based on the regular phylogenetic inference, based on either maximum likelihood or some Bayesian method, where you would first use a software to look at what is the best model that you need to use for the phylogenetic inference, and then go ahead and do the phylogenetic inference with bootstrapping, see what is the support for each of the nodes, and it's a very different approach. So if what you want to do is really infer what is the most likely or the tree that has the highest posterior probability, depending on the approach you decide to use, but your focus is a good species tree, then I wouldn't go through all this effort, because it's really not the best, it's really not the most likely representation of your species, but it's good enough for what you're trying to infer here with the orthology and phylogeny scenario.
Okay, and like using the orthogroups as an input for to do a phylogenomic tree with maximum likelihood or a Bayesian approach, that would be a good way to go.
I would say no, because then you would have something like what OrthoFinder already does. I say in that case, first of all, you need to know the group that you're going to do the phylogenetic analysis for, because there are different sets of genes that are informative for phylogeny, also depending on what you're including in there. If you have like very distant lineages and you're trying to do a phylogeny that covers, I don't know, tetrapods, which is a rather large and divergent group, you want to have a series of markers that give you the resolution at that level. If, on the other hand, what you're trying to do is like a species tree of a particular genus, which doesn't have that much divergence, then you choose a different set of markers. But this is specific to your biological system, and that should be the input. Like you could have concatenated multiple sequence alignment from all these markers that are relevant to your study system. But that's like an entirely different approach.
Okay, thank you. Thank you very much.
All right, and so what I was saying is I'm just gonna explain something very quickly. And for those of you who are already familiar with multiple sequence alignment and local and global alignment strategies, I mean, there was honestly not time to go into all of that, which is also like relevant conceptual background to have for this subject. I'm just going to touch on a distinction between what OrthoFinder does and MCL, OrthoMCL and OrthoLint do for selecting orthologs, which is actually, I think, very relevant. The first thing I want to say is when you use a local alignment algorithm, such as that is implemented in BLAST, you get several metrics that are relevant to this. I'm sure all of you have run BLAST and you know that there is a bit score and an expectation value. The first thing I mentioned first, the score of an alignment, just to build up on the concept of the bit score. It's basically the way to say whether your alignment is good or not. How good is my alignment? which is just a calculation of the sum of the scores of all the substitutions and gaps and exact matches that are present along the length of your sequences. And so say the score here, based on a substitution matrix that gives you, like it's given by the likelihood that those amino acids are going to be changed, or how often that occurs, like in real life, you get all the sum of scores of these individual positions, and that gives you a score of overall the quality of your alignment. So BLAST, that's something similar, but it's the bit score, which is just basically a normalized score that even if you used different substitution matrices to align each of your pairs of sequences, it's a score that you're able to generalize. Like you can compare a bit score across any of your alignments that were produced with the same set of sequences, but maybe changing different aspects of it, like the gap penalties or the substitution matrices. So the bit score, what gives you is an estimation of the magnitude of your search that you would need to do in order to obtain a score that is equal to or better than the score you actually observed only by chance. So this score is interpreted in a unit called bits, and it's just basically this normalized score of an alignment. But then when you run this BLAST type of analysis, you also get a significance value, which is the expectation value. And this is the e-value that you specify to say, what are the hits that you want to obtain in your search? And you usually say the typical is it's 1 to 10 times 10 at the minus 5. But the expectation value has some caveats because it depends on the size of the database you're comparing against. It also depends on the length of the sequences that you input as a query. This n is the size of the database. And you also have some parameters here, this k and this lambda, that depend on the substitution matrix that you use, the gap penalties, and the composition of your sequences. But basically what these equations relate to is that at the end, your expectation value changes really with the length of your sequence. And that as the longer your sequence gets, the more likely it is to obtain matches just by chance. And so any software for orthology detection that uses e-value as the cutoff is also biased by your sequence length, by your protein sequences length. And that is the case for OrthoMCL and OrthoVen, but is not the case for OrthoFinder. They do not use the expectation score. So I just wanted to make that clarity because that also impacts the differences that we will observe in the exercise between the two types of software. And I want to give the floor to Zaide, who is going to introduce the biological system and the example.
Okay, so.
Do you want to present yourself or you want me to pass?
No, you can help me with that, yeah. So, but I'm not sure if we are going to have a break here or after.
I think let's introduce the biological system, okay. And then we can really come back for practice.
Okay, so before to start our practical exercise, I would like to tell you a little bit about the biological system in which we are working on it. So maybe for those that don't know about insect chemical ecology, the insects use different cues, chemical cues, to do some specific behaviors. For example, some mosquitoes use different signals in the environment to locate specific places to lay their eggs. And other insects use different plant volatiles when they are looking for food. In the case of some insects that are hematophagous, they use like the skin volatiles, human skin volatiles, in order to recognize and to locate the host. The recognition of different chemical cues are really important for reproduction. For example, in most of the species have a specific species-specific interactions using pheromones, and in the case of some social insects, they use chemical cues to communicate it. The next one. So this is a picture of the insect olfactory system. So what you are looking is the head of a mosquito, and the mosquito has the antenna, and the antenna has some little hairs that are called sensilia. And in the sensilia, there are different pores, and the odors pass through that pores and reach that yellow part that it's called the sensilia lymph. And in the sensilia lymph, we can find different types of proteins. So the first groups of proteins are the odorant-binding proteins. And one of the characteristics of these proteins is like these proteins have this signature that is a conserved pattern of 6 cysteines. We have a second group of proteins that is the odorant receptors, and the signature to recognize them at sequence level is like this group of proteins have 7 cross-membranous domains. We also have other family of proteins that are the ionotropic receptors. And the last family are the gustatory receptors. So all these proteins are involved in the detection of volatile compounds. So when we are talking about the functions, they made the same. But in the next slide, we are going to work with some data from odorant receptors. And there are some specific odorant receptors that are involved in the recognition of pheromones. And in the case of a Lepidoptera group, that is, you can find the butterflies and also the moths in that group, there is a specific clade in which all these type of protein are grouped. So this is like an ideal ortholog group to work on it. So something that is also important is like from previous studies, we know what is the role that have some of these proteins. So we know what is the specific volatile that a specific proteins can recognize. So we are going to use some sequences as a positive control because we have experimental validation just to at the end to our comparison. So this cluster is a single lineage, as you can see in the in that part, in the other part. That this protein family is specialized in the recognition of in a specific volatile, and this family.
Sorry.
Some other, yeah.
Wait, you were muted for a second.
Yes.
Can you both go back 30 seconds?
Okay, I was saying like this lineage is specialized in the recognition of a specific volatiles that are the pheromones, and it also have another signatures that allow us to recognize them at sequence level. So this is like an ideal data to do our exercise because we have a lot of previous knowledge about them. So in the next slide. Yeah. So maybe we can make our break here, and then after that, we can just start to do all the practical exercise. The break will be 10 minutes, I guess.
Also, in the meantime, we know that right now it's maybe lunchtime or dinner time for some of you, depending on where you're joining from. So we want to give you some time to like eat something or just grab a quick bite. But also, maybe we can use some of this time to make sure you're more or less up to speed with the installations. So just take a bit of time to take it slow, just eat as you go, and just like regroup later to start the analysis. But if you had any issues with the installs and so on, maybe we can talk about that as some people go for a break.
So how long it will be the break?
10 minutes could be.
10 minutes. Okay.
Yeah.
Good.
I will be here. If someone have a question, I could be stay. If you have any problem during the installation of the program, you can just ask me, or you can write in the chat.
Can you give us, please, like an installation link or?
I don't know if you receive a mail with all the instruction. We made like a manual. And the installation takes more or less 20 minutes or 30 minutes. So
Lucy, you guys did share all of that in advance, right?
Yes. Yes. We send all the information by email.
Okay.
Did you not, someone here did not receive that?
Yes, I received the email today, so I don't have the program installed, but I can see anyways the I don't know, between you do the exercise, I can see in the screen.
We also share a video with the tutorial.
Sophia, I sent you the mail with the material with the link today.
Yes, but today, so I don't have the program installed yet.
Okay.
Okay. Maybe Zaide, can you share the files with the proteomes?
Yeah, I will share with you.
Maybe even the pre-computed results with Lucy, with all the organizers.
Yes. If you
so
want.
that you send it out to all of them during this break. Because we do have pre-computed results so that you can navigate through them. Also, if you do not have a Unix-based operating system, you cannot run OrthoFinder. In that case, you also need to see the pre-computed results, which you will be able to navigate in any type of operating system because they're text files basically. So it was important to have these pre-computed results anyway. So we'll send them to you just in a minute.
Yeah, I was looking through my mails and I didn't receive the one that you were mentioning. I already have OrthoFinder installed, but I don't know if we need something else.
Could you please send your email or write your email in the chat box so
Yes.
we can check the information? Thank you.
Hola, también tengo una duda con la instalación. Logré instalarlo en un ambiente de Conda, pero estoy viendo en la instrucción que el DLC par, ese no se instaló como dependencia. ¿Lo instaló aparte o ya viene ahí?
For a basic running, you don't need it, but maybe you can just install later, but for our exercise, it's not necessary. And we also are gonna give you the pre-computed results. So, but if you wanna do all the inference of the gene trees and the species trees, you need it.
Okay, thank you.
The DLC is short for duplication, losses, coalescence, and that is the software behind the connection of the second part of OrthoFinder 2. Well, you need all this inference and reconciliation. So if you want to go all the way to the second part, you need them. You need them.
Okay.
But not to generate the orthologous and paralogous groups. This is not necessary.
Okay, thank you.
I will share the link here in the chat for everybody.
Ah, it's better, yes. Claudia, do you have any question?
Maryam, can you help me to check if it's working properly, the link?
Yes, it's
yes,
working.
I can access it without problem.
Yeah, me too.
I'm not sure if I understood. I downloaded the DLC part. In your tutorial, you said that we must like install Python in our directory, something like that. DLC just works with Python language. I don't know this.
No, when you are doing the installation, they just do that automatically. You don't need to install Python or something like that. You need it.
Okay. Mm-hmm. Oh, sorry. Thank you.
You're welcome.
There is a comment from Victoria on the chat.
I don't see it.
Because maybe you are using the option the diamond and not blast option. Okay, could you just show me the error? Okay. Okay, you are typing wrong. Let me check.
But Lucy, was it only Sophia that received the email today?
Yes, because
Yes.
she sent the registration today.
Okay. Yeah, because this wasn't sometime in advance.
Yes, like a week ago or something like that. Yeah.
Okay.
Well, in any case, that has the video with the instructions, and the idea is that we'll focus today on the analysis and interpretation, because solving all the install difficulties right now, it would eat up the time and we won't be able to move forward. But you will have the tutorial not only for the install, but also to the post-processing, let's say. And you can do this on your own time also later on and come back to us with questions. You will have our emails in the presentation. So yeah, no worries. You'll be able to, the idea is that here we focus on understanding it and not the difficulties, practical difficulties of installs and that stuff.
Yeah, I'm going to. I don't know if you again are using apt.get install, and you have to use apt
dash
get nor dot. Maybe that could be because you are using dot and not dash. Because it's the only thing that I see.
Adriana, what operating system do you have?
Oh, Fedora.
I'm not sure about Fedora. If someone else has experience with Fedora here, because I use
No, me neither.
Ubuntu.
Let me check.
Victoria, it seems that either you have not installed Diamond, which is something you have to install additionally before OrthoFinder can find it, or you did not add it to your path yet so that it can be found when OrthoFinder is called.
I haven't received an email, but I already downloaded the files that Zaide shared in the drive, and I have OrthoFinder installed, so I don't know if I need anything else. Okay, perfect, thank you.
Maybe we can create another space, so then you can just send me an email with the errors and I can help you later, because we don't have enough time to solve all the problems now. But I don't know if you download the data that we share with you in the link that we sent previous. Because the idea, like, let's
Can
try
you
to do.
share your screen, Zaide, and you show them where the data is in that shared folder?
Okay, let me show you very quickly. So here, it's like in the chat there is a link that I sent you, and the idea is like you have to download this folder. And in this folder, you will find these files. You will find these files to do our exercise, and you need to download it and storage in a directory, all of them.
So these are the predicted proteins for 10 species. What do they contain, each of them?
Okay, so each file, we forgot to share the PDF with the exercise.
We can, I'll put it in the same folder.
Yeah, please. So each file contains olfactory receptors per each Lepidoptera species. And we also identified and we use like a signature for those receptors that has been previous characterization, and that kind of proteins are going to be our positive controls. So the idea is like just first download the data, and then I will show you like the relationships between these Lepidoptera species. Okay, so now in the link that I sent you before, you're gonna find, I will show you. You will find this this PDF, and here is all the explanation of the data that we are gonna use, and also the instructions. I don't know. And here is like also again the command that you have to run, but using the data that you are gonna download from the link.
So as you see, it's basically one command, and it's very quick because this is a test data set, which is small. Yeah, so it's only that one link. And for OrthoVen, we just do it directly on the web, which is basically also inputting all the proteomes. And you know, this general workflow that I showed you when we went through the OrthoMCL pipeline, just basically always the input is just the predicted proteins of the species you're interested in. And that just initiates the comparison of all genes against all genes. And based off that, then it's the creation of the orthogroups depending on what each algorithm is using. But it's a basic input. And in any of the analysis, that's the starting point, basically.
So I would like to know if you have already downloaded the data and if you are ready to run the program. Claudia? Do you have a question, Claudia?
No, I was just saying that I'm
You're
ready.
ready.
Yeah.
You're ready, okay.
Yeah, I'm ready to.
Okay, very nice. Something that is really nice with AutoFinder is like you can use a very big amount of data and you will have the results quickly. And after we run our practical exercise, you wanna see that the output folder that AutoFinder create is really good. Like you're gonna find like all the information very organized. So I don't know if the others are ready to do the exercise, or probably you already don't or run the command. How to parse? What do you mean to parse output from orthologous?
So like the R command, you have to visualize the
not with BioPython, but you can use, we are going to show you how to use R, but there are another programs that you can use. You can use Cytoscape, and also Orthofinder will provide you a folder with the gene trees and the species trees, so you can just upload that to iTool or another program to visualize the trees.
Mm-hmm.
We are going to give you an example of a specific file that could be really useful to visualize the relations, like at the orthogroups that are overlapping between the different species, but using R. We don't have an example with BioPython. Oh, no.
Well, since it's only that one comment, I think we can go into the results.
Okay. Also, one of the participants have already run the command just to verify that it's working properly. Because yeah, it's just one command. Yeah, Monica. Okay.
Do you want to share your screen just to show how you?
Or you can share with us a screenshot, because it's just one command and that's it. And I will give you some recommendations. I will show you some tips that you have to have in mind before run the program. Monica, Jessica, could share.
Who is going to share, Monica? Because I need to give that person permission.
Permissions.
So who?
I don't know who wants to share the screen. I can, Monica. It's the same as the example test. And as you can see, thank you. She just used a one-line command, and then she just wait, I don't know, for just less than a minute with that number of data. And can you show just the folder that Orthofinder create? Thank you. So something that is really nice, that I mentioned before, is like Orthofinder. It's gonna give you all the results in a very organized way, so you can explore all the outputs. And we are going to show you how to use one of the files that Orthofinder will create. Thank you, Monica. It was easy for you? Musical. Someone else could run the program? Okay, so maybe it's time to see what are the different folders that Orthofinder create.
If you could not run it in the same link that Zaide shared before, there is the Lepidoptera dataset, which is the input for the analysis. But then there's also a results folder from last month, which is when we were preparing, but it's basically the same output structure from Orthofinder. So you can download that and follow us during the explanation of the output and what is contained within each of the directories. So if you want, you can do that too, in case you either have installation problems or have an operating system that just doesn't support running Orthofinder.
Yeah, okay, so I'm going to share my screen to show you the results. Okay, here. So let's, in the PDF that we share with you, there is at the end of that PDF a script that you can use to get this kind of figures. So I would like to start with this one. So here in all the axes that you will find like the different species that we use. And also this is the number of ortholo groups that are overlapping between each species. So this is like the ortholo groups overlaps. This is like another way to see the results, and this is really similar to an ortho Venn diagram or Venn diagram. So yeah. So just to tell you a little bit about the results. So the first one is like when you go to the ortholo groups text file, you will find that the first ortholo group have 7 of our positive control sequences, and we were expected that all of them were in the same cluster because all of them made part of the same lineage, as I show you here. Like we were expecting that all the positive controls were in the same lineage, but in this case that doesn't happen. Another important thing, like the proteins from the same species and not in the same cluster. That is we were expecting that if we have sequences that are from the same species, we were expected that all of them were in the same plate because they have a really close relation. But when we include in another analysis more data, as we mentioned during our presentation, we found with AutoFinder better results that I'm going to show you here. So in this tree, we use 800 proteins, and we found very like expected results, and it's just because we include more data and we also include more species from the same order. For example, something that I would like to show you is like when we were doing the example exercise that you have in your computer now, in your files, we couldn't get this type of plates, like not all the sequences from the same species were together, but when we include more data in our analysis, we get better results. So this is something that could be, I don't know, important when you are doing your analysis. If you have the opportunity to include more relatives from the same group that you are analyzing, probably that you will have like a results as you expected. And something that is important, like don't forget to use all your positive controls just to know if you are getting or not the proper results. Okay.
So maybe I can show for a moment just how this would be done quickly online with Orthovan.
Orthovan, yeah, sure.
Before we go into why are there differences?
Yeah.
All right. So quickly, I'll show 2 things. So the first one is that in the practical exercise PDF that we also share with you, there's a detailed explanation of the results files. for OrthoFinder, which basically contains all the ortho groups where each row contains the genes belonging to a single ortho group. Then you have all the genes that could not be assigned to any ortho group. This is the basic output format that you would obtain by running MCL, for instance. You also have these single copy ortho groups that actually, this is what I mentioned that is very important to, like these are the ortho groups that contain exactly one gene per species. And these single copy ortho groups are used for species 3 inference methods. So this is a useful output to have. So Claudia, you could use, for instance, this input maybe if you don't have a very well conceived like prior to know what are the good markers to include in a phylogenetic inference analysis, like separate to this, to produce the species tree. Maybe you can start with this output, for instance, but you can also get the gene counts for each species in each ortho group. So basically how many genes for species A, B, and C belong to ortho group 0, 1, 2, 3. You also get the statistics calculated from the ortho group analysis. You have statistics overall or per species, like on a species by species basis, and you also get a calculation of species overlaps. And this is what Zaide was saying to produce the heat map analysis. This is based on this last output that I described right here, which is the orthogroup species overlaps. You can use that file to run the script that Zaide provided. And this is in the email that was sent in advance about a week ago. We also asked you to already have installed these 3 packages. And this is a very, very quick script just to basically produce the heat map that she showed us in the presentation. And the way you do it here in Orthoven, this is, let me copy this link out to you in the chat box. Basically, you go there and you click on start, and remember 2 things, that Orthoven works similar to MCL, and it works using as a coding threshold value the expectation value of the all versus all comparison. So you can become as strict as you want with the expectation value. Let's use the usual one. And this is the only other parameter value that I mentioned before, which is the inflation value. And you can do 2 things. Either use the groups that they already pre-computed, like there are several different kinds of taxa that already have a pre-computed output in Orthoven 2, fungi, bacteria, metazoa, well, a lot of different things. For them, they have like different features because these are like well-known and annotated genomes. But then you can also upload protein sequences that you have for your own work. And this is where you'd click to start your analysis. So you have to upload a FASTA file for each of your species 1 by 1. So basically, if you go to a Lepidoptera data set that you just downloaded from that link, say this is the first FASTA file, you drag it here and you give it a name. So Agrotis sergatum. And then add to analysis list. And you do that with each of them. So in this case, you do that 10 times for each of these FASTA files that you have in your input. but I'm not going to do that here because that takes a bit of time. So basically, this is the output that comes out. There are, so here you have the shared or orthologous groups that were formed in the analysis. These are the 10 species. And what is highlighted in the green, in like darker green, is just basically which are the species sharing those clusters. They're called here clusters. And so there are 3 clusters that are shared by all species, which is the top cluster here. There is one additional cluster that is shared for all except for this Australian nubialis, and there are things that you would expect to find. For instance, there have to be orthologous groups that are specific to the same genera. For instance, all these Spoloptera share these different orthologous groups. The Australians should also be sharing something. The reason what they show here in the bottom is that the orthoven automatically, like the order that what it uses to show them are based off how many species share a particular set of orthologous groups. But you get that for the bombyx, or just if you have this structure where in your sampling strategy you had species of a particular genus, you should be able to observe that in your data. Since we do not have all the proteome of all these species here, then that's why this isn't an exact reflection of what you would observe for each of the species if you did have all the predicted proteomes for each of them. But for the purposes of like demonstrating the capabilities, the quick analysis was what we prioritized. And for each of those clusters, you have associated a protein count, and also you can download a summary of the amounts of proteins and clusters you have for all of them. And you can also generate the same types of Venn diagrams that you can do in R for those two, but only here you can pick 6 species at a time. So if you wanted to look at other combinations of species, you have to select and deselect some of them. But of course, since we used a low number of proteins per species, then that's why you don't see here like larger numbers. There's another, so you can also get the pairwise heat map, which is the analogous of what Zaide just showed. It was the output of that R script. You can get the abundance count of the number of proteins that are shared overall between species. Of course, you expect that Mori and Mandarina, which are 2 species from the same genus, will share larger numbers of proteins compared to others that are not. Which is basically, this gives you like a well visualized output of the orthology relationships across species. And there's an additional feature here. Let me see if I can find it quickly. So this is the same file of the orthologs that you get also in the orthologs output for OrthoFinder. But let me go back. Clusters. This is the legacy kind of output of all clusters, and each lane is 1 cluster. And let me see if I can get to. I want to get to, maybe we cannot get that feature here. But when you use the species that are well annotated, you can also observe the annotation, functional annotation of each cluster of orthologous of orthogroups. And just have that associated like to the Swiss prot ID and things like that, which is something you can get if when you click start, not here, not here, here, here. Why won't you click there? I don't know what's going on. But basically, when I click start, remember that we had these tabs, metazoa, plants, bacteria, fungi. If you use the examples, or if you happen to work with species that are well studied and well annotated, and you use the species from this set of pre-computed examples, you can also observe the functional characterizations of each of the clusters, which is cool to have. Maybe right here. So mostly you won't get, so for each of the clusters that they share, see, this is a signal transduction notation for the Go terms, and you have the associated Swiss prot hit for each of them, each of the clusters. But in this example, we won't get a functional notation for the majority of the clusters, but you will, that's what I meant, that you will if you use any of the well-known pre-computed examples for those groups. Right, so then we can go back here. So this is just for showing you some of the results. And so what I just wanted to say is that the precision of OrthoMCL is much, much lower than what it is for OrthoFinder. And that is a big reason why the algorithms give different results. And also because under MCL, when it's not corrected with all the different features, like what OrthoFinder, like what I mentioned before, is not based on e-value, all that, it has a bias, which is that short genes are under clustered and long genes are over clustered. And that is a result of using the expectation value. And so that's something that is heavily going to bias your result. And so, well, I just wanted to say that that was a reason behind it. But I wanted to know if you guys have some other questions on the outputs, if there's something you're not understanding before we move on to more like the conclusion section.
So here, so again, someone have any question or wants to know something else about the softwares, about how to run the program? If not, okay, so we want to give you like some measures to have in mind when you are going to work with an orthology inference. So when you are using OrthoFinder, so OrthoFinder infers orthologs from the orthogroup trees. So remember what Maryam explained you, like they not just use a gene species, they also use species trees. There is something that is really important when you are running other kind of programs like OrthoGen or OrthoMCL. Is like you have to you have to have in mind, like the length of the sequences could create a bias, but in the case of the Ortho Finder, this program helps you to avoid that. And the last part is like as we mentioned during the presentation, Ortho Finder takes the information when they are doing the phylogenetic approach to provide additional information. And when you have time to explore the folders that we give you with all the research, so you will find like a folder with all the rooted gene trees, with the rooting species trees, with all the information about the gene duplications, with the ortholog groups, with the paralog groups, with the imparalog groups. So this is really nice from OrthoFinder. Another thing that is important when you are running the program with your own data is like, don't forget to rename your sequences. If you see the data that we provide you, like all the headers of the sequences is really short. So before to do the running with your own data, do it this, because if not, you are gonna Ortho Finder, it's gonna work properly if you have a big name of your sequences. Don't forget to use positive controls. If you can include data that have experimental validation, do it, and also try to do use a signature to identify them quickly. And if you want to know what are your positive controls and in which group they are state, so you can check this file that is orthogroups.txt. And don't forget to use all the files in the FASTA format. And if you are gonna compare different types of species or different organisms, you have to have one FASTA file for each type of species or strain, or depends on the organisms that you are working. And at the end, with the results that OrthoFinder create, you can also use different tools, softwares. You can visualize the results with R. You can upload all the gene trees or species trees to iTool. You can also convert the trees to networks, and you can use another software like Cytoscape. At the end, what I want to say is like this software is really flexible, and it will give you a lot of information in the results folder that you can just play it and try to extract a lot of information for your analysis or for your research. And this is all the workshop. We hope you enjoyed all the course, and if you have any questions, you can contact us. Here are our emails. If you have any question in this moment, you can just do it. If you want to know something else, feel free to do that.
And now for the end, well, just thank you, Zaide. Thank you, Maryam. It was really nice. Yes, and we want to take like a picture for all the participants. So we want to invite you to open your cameras, please. We want to know who was here, who were here. Okay, I'm going to start.
Did you just take the picture and didn't let us know?
No. Not yet. I'm going to do it. I have to take 2 pictures because we have a lot of people. Talia. Okay. I have a lot of people, so we have like 2 to text. So please smile. And the second space is smile again. Okay. This is all. Thank you so much for being here. We are very, very, very happy with this workshop.
Yeah,
Thank
for
you.
your time, for your
Get in
attention.
touch.
Thank you.
Okay,
Thank you.
bye.
Thank you. Bye-bye.
Thank you.
Thank you.
Thank you, girls. See you next time.
Bye.